Hello, everyone. I would like to welcome you to today's webinar prediction of prime editing insertion efficiencies using sequence features and DNA repair determinants. My name is Gemma Garcia. I'm the Regional Marketing Manager at Bioscience. So now I want to introduce you the speakers for today. Jonas Kappen is a fourth-year PhD student, also in the laboratory of Leopold Parts at the Wellcome Sanger Institute in the UK. Jonas uses CRISPR prime editing to insert recombinase recognition sequences into the genomes of mammalian cells to trigger large-scale genome rearrangements. To further develop the prime editing technology and build predictive models to guide the choice of optimal reagents, he characterized the insertion efficiencies for thousands of short sequences using whole CRISPR prime editing screens in different cells, cell lines, and repair contexts. We also have Juliane Weller. She is a third-year PhD student in also the laboratory of Dr. Leopold Parts at the Wellcome Sanger Institute in the UK. She uses CRISPR prime editing to introduce genetic variants in vitro, including deletions. To better understand the determinants of prime editing efficiency and guide experimental design, Juliane has been building predictive models for short sequence insertions. They are based on editing efficiency rates of thousands of short sequences from pooled CRISPR prime editing screens at different target sites and in different cell lines. Juliana, you can start.
Hi, thanks for having us today. We're going to talk about inserting sequences with prime editing. So we're going to show you the experiments we did in the lab to insert lots of short sequences into the genome, the features we identified, and then the tool we built to predict insertion efficiencies. The data has been published recently in Nature Biotech, so if you want to have more details, just check out the paper. And looking back at CRISPR technologies and how they evolved, it started around 11 years ago with CRISPR Cas9. Originally, CRISPR was a defense mechanism in bacteria and really disrupted the field of life sciences when it was discovered that it could be used as gene editing technology. So you can see CRISPR-Cas9 is molecular scissors. They find the target site in the genome and then cut it, creating double-strand breaks. During the repair of those double-strand breaks, small insertions and deletions can be made, which then disrupts gene function. But we can also use the Cas9 as the search function of Cas9 to link other enzymes with it. That has been done for base editors. We can also consider them as molecular pencils because they can do substitutions in the genome. And here the aminase is linked to that Cas9. So that helps identifying the target sequence. And then the deaminates the bases, which then leads to a conversion of them. The newest technology is prime editors. So here the Cas9 is linked to a reverse transcriptase, which then leads to a search and replace mechanism. So we can consider them as molecular word processors, where you can find its target sequence and then write in very precisely a new sequence. And prime editors can be used for insertions, deletions, and substitutions. So they're very versatile and very precise. And we were really fascinated by prime editors. So we wanted to understand better how they work, and in particular for short sequences. Short sequences are interesting because you can use them to generate gene knockouts, for example, by inserting a 4-nucleotide sequence that has a stop codon, but also introduce a frame shift. Then we can use short sequences for protein labeling with fluorescent proteins or purification tags. We can insert landing pads that can then be used to later switch in larger genes or gene fragments with recombinases. And using short insertions, we can repair pathogenic deletions, like, for example, for cystic fibrosis, which is often caused by a 3-nucleotide deletion. In the first example, I want to just talk more about the protein tagging. So imagine you want to tag a protein here after the ATG. You have different protein tags you can choose from. And for some of them, you might even have different codon variants where some of them have different editing efficiencies. So it would be nice to have a tool that can tell you which codon version and which tag is most likely to insert well. Looking at prime editing, I mentioned earlier that it's a nicking Cas9 that is bound to reverse transcriptase. So the nicking Cas9 generates a small nick in the DNA, and then the reverse transcriptase can be used to directly write in the edit into the genome. And what we also need for that is a pegRNA, a prime editing guide RNA. Similarly to the Cas9 guides, it has a guide area where that is homologous to the target region. It has a backbone, and then we have here the 3' extension where the template for the edit is. The 3' extension also has a primer binding site, which hybridizes with the created flap in the DNA. And then the reverse transcriptase synthesizes the edit, which leads to 2 DNA flaps. And depending on which flap is integrated and which flap is repaired, we either get the edit in the DNA or the DNA is like put back into the original state. So having a closer look into the pegRNA, I mentioned we have a spacer, a scaffold, a homology arm, which is part of the target sequence after the spacer, inserts, and then the primer binding site. And we looked at inserts from 1 to 69 nucleotides. So build a library of over 3,600 pegRNAs. They included utility sequences, eukaryotic linear motifs, sequences with different structure, and a lot of short sequences, up to 10 nucleotides. We then tested the insertion efficiencies of those inserts into 4 target sites in the genome using 2 different cell lines, the Hector-9 T cells and the HAP1 cells.
And so maybe first a couple words to the cloning. So this was what Juliana showed you was the design of our experiment, but how do we actually make a screen? And it's actually somewhat tricky to clone pegRNA libraries because you see a pegRNA consists of all these individual parts. And you have a variable part, which is the spacer that finds its target in the genome. Then you have something constant, which is the scaffold. And then you have a variable part again that directs the edit and has homology to the target. And in addition, if you would then go for a enhanced pegRNA, you would have something constant again. But we had different strategies throughout the manuscript. And the first one we did is we did a 2-step cloning where because we only targeted 4 sites in the genome, we could first generate these vectors that target 4 different places. And instead of having the insert library in the vector, we added in a stuffer sequence that's flanked by restriction enzyme sites so that then we could go to Twist and order an oligo pool that contains all these different inserts. And then these inserts would also be flanked by restriction sites and some primer binding sites and just some kind of filling sequence to make everything the same length. And then we can amplify it. We can cut it out and insert it into our entry vectors. And so in this way, we got a library with all these diverse sequences. So this is how we did the first screen. However, then we ended up doing a lot of validations. We did smaller libraries. And what we noticed actually works reasonably well is to directly order the entire thing on one oligo. So there's the option to order up to 300 base pair oligos, and then we chose those ones where you can put the spacer, the scaffold, the homology, the insert, and the primer binding site all in the same oligo, which also led to slightly more uniform libraries. Yeah, so this is how we made the libraries, and how did we perform the screen. So we had at first an antiviral guide RNA libraries. We put them into cell lines. We selected the cells that got a pegRNA using blastocidin. And then for the Hector-9 T cells, we transfected in Prime Editor transiently, and we could select for the transfected cells using pyrimidine. And then after 5 days of editing, we harvested the cells, extracted the DNA, and read out the edits in the library. And for HAP1 cells, which were our other cell line model, which we chose because they are mainly haploid, which means they only have one copy of their genome. So we think it's a very useful cell line for a lot of tagging and this type of studies. And since they're harder to transfect, we instead first generated cell lines that already stably express the prime editor, which was introduced using a piggyback transposon vector. And then we could just induce the expression of the prime editor, doxycycline, wait 5 days, harvest cells, and again, extract DNA and perform next-gen sequencing. So how does the data actually look? Here, every dot represents one insert sequence, and you can see that it's there is quite a big difference between the efficiency with which one insert is made, and you can see it's concordant across replicates, and yeah, it spans around 100-fold difference. And also we had context, so we had different cell lines, so we could see how does it compare across different cell lines. And you can see that the correlation is less than for replicates. And specifically, there's an island of sequences here that stands out. And that island of sequences are short sequences. And we talk a little bit about those in a second as well. Then we could also look at the same cell line, but we had different target sites. And again, we see a drop in correlation compared to replicates. And finally, we had the lowest correlation when both the cell line and the target site were swapped. But what explains this? So we see there is some reproducibility of the insertion rates, but also some difference based on context. And one feature I'd like to show you is the length of the insert sequence. So you can see here, every dot again represents the editing rate of one insert sequence. And you can see that short sequences are made efficiently. Then there's this drop in insertion rate at around 14 nucleotides. It goes up a bit again, and then it flattens out and gets lower for the longer sequences. So length explains why some sequences are made better than others, but it also explains differences we see between cell lines. Because if we compare the HEC293 T cells and the HUB1 cells, you might notice that the biggest difference is for these short sequences, which are made much less efficiently in HUB1 cells. And by now, a lot of beautiful studies have shown that mismatch repair is one of the main drivers of insertion efficiency of short sequences. And HEC293 T cells are mostly deficient in mismatch repair, while HUB1 cells are a wild type for mismatch repair. So we thought, okay, if we knock out mismatch repair, does it recreate an insertion profile similar to the one in HEC293 T cells? And in fact, this is what we see. So now we see that this library works pretty well in looking at the effect of something like mismatch repair. So we thought, let's go through the entire prime editing pathway and test out the contributions of a lot of different steps, such as transcription, the reverse transcriptase, the prime editor protein used. And we go into a lot more detail there in the manuscript. But maybe one of our more surprising findings was the influence of exonucleases, and we'll talk a little bit more about them here. So the prime editing creates these flap intermediates, and flap nucleases, which are specific either for the 3' flap that contains the edit or the 5' flap that contains the wild type sequence, could potentially interfere with the editing process. And to test out their direct contributions, we cloned these constructs and overexpressed them together with a library targeting the HEC3 sites and HEC293 T cells. And you can see that if we overexpress EGFP, which is our control, and compare that insertion rate to cells without overexpression, you can see that it's quite tight overall. And we also see that same tightness when we overexpress FEN1. It might have some difference on some sequences, but overall it looks relatively consistent. However, when we overexpress TRX1 and TRX2, the two flap nucleases that could digest away the flap that contains the edit, you see there is a strong difference between those and no overexpression, where short sequences behave similarly, but the longer sequences are made much less efficiently when the nuclease is overexpressed. And you can see it more visually here, that while short sequences are still made, the longer sequences are very suppressed in these overexpression contexts. And our hypothesis was that the reason this works is that, or the reason that we see this is that the nuclease can digest away the flap. And we have some data that points towards this. And we can make use of a little trick. So the HEC3 site that we target in HEC293 T cells actually has a SNP. So it's a single position in which the nucleotide is different between the alleles. And also HEC293 T cells are very far from normal cells. So they're triploid here. And you can see that 2 alleles encode a G and 1 allele encodes an A. So you can imagine that if the prime editor edits that allele that has the A here, but our PEG RNA contains a G, that would create a mismatch. And if the entire flap was integrated, you would expect that the edit contains a G here. But if the flap was for some reason degraded to shorter length and then it ligates into the genome, you would still see the A. And we can quantify that ratio of A to G for reads that contain the edit. And what you see is that without any overexpression, we have about 1% of reads that contain an A in this position, which of edited reads, which means that the majority had flaps long enough that the G got fixed in. However, and it's very similar if we overexpress EGFP of N1. However, if we overexpress TRX1 and TRX2, you can see how that fraction of reads with an A in that position goes up. So that could point towards something where the flap starts becoming degraded more with overexpression of these nucleases. And then Juliana and I will walk you through a couple more smaller features in our set. So one of them is looking at the nucleotide content. So you can see that for the HEC3 side, here we have the fraction of each nucleotide in the insert sequence and how it affects the editing efficiency. And you can see that having cytosines in the insert seems to be beneficial, while having adenines and thymines seems to be bad. And is it consistent? So we looked at our different cell lines, different target sites, and also some validation follow-up target sites. And you can see that it seems to be that cytosines are always beneficial in this position, and adenines and thymines almost always a disadvantage, although there's some variation between the individual sites.
So as Jonas mentioned, those are a few features we identified. We can summarize them into sequence features. So we saw that nucleotide composition plays a role. The insert length is very important. Then we identified structural features when analyzing the data. I'm going to talk more about the RTT structure later, but also pairing between the insert and the spacer, and system features like the exonucleases and mismatch repair that Jonas mentioned, but also having an intact backbone loop and terminator motifs in the insert. So let's get to prediction of insertion rates. We took all the data from the screens we had. And so basically we had different features, we had all the inserts and the measured editing rates, and trained an XGBoost model on that. And this then leads to the tool MinSpy, modeling insertion efficiencies for prime insertion experiments. And then we tested the prediction scores on our held-out test data set. So here we want to see how well does our model perform on insert sequences it has not seen before. And you see the correlation between the observed and the predicted insertion rate here. So we got an R of 0.68. We also tested the model on external data sets, but I'm not showing you the data here. The model can be used to understand how computationally the score is predicted. And here you see the different features we used, sorted by their feature importance, and then a value that indicates how it impacts the model. So if it's a positive Sharpe value, that means that the model predicts a higher predicted insertion efficiency, and negative Sharpe values means lower predicted insertion efficiency. If we go to the example that Jonas gave earlier with length, where we have like this peak, so very short inserts are highly inserted, we can see here that inserts with a low feature value for length, so short sequences, have a higher predicted insertion efficiency. So that matches well the observation that we had when analyzing the data. Another very important feature is the RTT structure. When thinking about the structure of the RT template, so this is the insert and the homology arm, but also we can think about the whole extension of the PEG RNA. There are different ways of calculating the structure. So initially, we just looked at the impact of the structure of the insert alone. But we can also think about looking at how insert and homology arm interact, or how the whole extension folds up. There was a paper where they protected a 3' end of the PEG RNA, called it ePEG RNA, and that protects it from being degraded. So that increased the overall availability of the PEG RNA. But that's not what we're looking into here. Here we're really looking at structure inside the PEG RNA. And we identified the structure of the RT template being most important. So if you look at the correlation between the calculated structure, so the free energy of the insert in the homology arm, and the insertion rates, we see a very clear trend that stronger structure increases editing rates. And our explanation, or like what we think could be happening, is that with TRX1, TRX2, structure can protect the flag from being degraded. If we look at our overexpression experiments, we see that structure is important for all different insert length categories, but particularly important in the case where we have TRX1 and 2 and for long inserts. We can see this in the model with insert sequences that have a low structure, so low structure values, low energy, meaning strong structure being inserted more efficiently. Then MMR proficiency is another feature that we talked about earlier, but you can also see it in the model. So being MMR proficient means having a high feature value for MMR proficiency, and in that case, it can be it can lead to a negative impact on the model output, so lower predicted efficiencies. Now I want to demonstrate you the tool that we built to predict insertion rates. So there's an online tool that you can use where you can input a target sequence and the editing position and the sequences that you would like to test. And you can also increase the length of the homology arm, for example, choose a cell line and add the mismatch repair proficiency, and then get a predicted value for it and see it on a distribution of different measured insertion rates. So like some sequences are insert better than others, and you can use the tool to test your own sequences. And also then export the data on a CSV. Now getting back to the example from the beginning of the HIS6 tag. We used more of it, measured more protein tags and predicted them to use the tool on them. And we can see that the sequences that we predicted to insert poorly indeed insert at a lower rate than the ones that were predicted to insert well, that you can see here in red. Another thing we observed in the data was that was this weird bump, and we were wondering, can we use that and pad sequences to increase their editing efficiency, so making them a little bit longer. We tested that for 3 different sequences and predicted a good padding for them. And indeed, padding them increased the observed insertion rate by 1.4-fold on average. So to sum up, we can see that prime editing is a very versatile tool because it can do all kinds of edits and has a high precision and doesn't create double-strand breaks. But insertion efficiencies vary. It mainly depends on insert length, structure, and insert composition. But also the repair pathways in the cell are very important, in particular mismatch repair and TRX1 and TRX2. And you can use MinSpy or the data that we have in our publication to choose efficient inserts. So you can rank potential inserts by their efficiency and choose between protein tags, optimize codon composition, and predict padding sequences to insert to increase the insertion rates. And with that, I want to thank the people working with us on the project, in particular Leopold Potts, our supervisor, and Ellen, who has been running the experiments with us, Fabio and Anand for helping and supervising, and Ivan, Hedi, and Uku for the website development, and all the other people in the lab and that we discussed with. Thank you very much, and we are happy to take questions.
Perfect. That was really nice. Good. So it seems that we have some questions. Okay, first one, is there a reason why you choose the path of overexpression of TREX1 instead of a knockout approach?
Yes, so we had a couple of thoughts there. One, there's TREX1 and TREX2, so it would be, and we weren't sure. We thought that maybe there could be some redundancy. Also, we didn't know which nucleases could be involved. So I guess we were doing it in a relatively small scale. So it was more of a maybe lucky shot type of experiment. And we had some idea that Trex might work because it would make sense from the mechanism. We also had a chat with Brett Adamson about this. And there was one thesis out there that showed it to some degree. But yes, so I guess if you want to knock it out, there could be that it has an effect, but you don't see it because it's masked by the other nucleases that are still around. But we thought that if any of them have any effect, if we overexpress it, we would most likely see the effect. So that was the main reason. And then maybe another reason that it's technically a bit more tricky to knock it out because our cell line already expresses prime editors. So we could, I guess, come in with shRNAs or do a CRISPR-I first, as in the repair-seq paper. Yeah, it would mean a couple of editors that we need to throw in, but here we just needed to put in an overexpression construct.
Okay, the next one says, as the model focuses on predicting insertion efficiency across full target size, the different protospacer would be involved in the dataset. Would it be useful for prediction of protospacer first?
Yeah, that's a good question. So if you want to design your own experiment and insert sequences, we would suggest to first identify a good target site, and you can use that, for example, by using Predict, another prediction tool for prime editing efficiency that is focusing on a lot of different spacers. And or you can use DPE, another tool, to find a good target site and then predict different insert sequences with MinSPy. But we've seen when we validated the model, I think I can show the data for that, on different target sites. So for example, on the DPE data set, that for most target sites, the effects that we've seen for insertions like length and nucleotide composition still play a role and still hold.
Good. Okay, we have a lot of questions. I don't know if we will have time to answer all of them. Okay, so regarding the software that you showed, the MinCP, I don't know how to pronounce that, sorry. The question is, what is the good versus the bad value? Are the tails of the distribution higher efficiency?
Maybe as a good or bad value, we usually predict relative insertion rates. So we score an insert against our library. So a good value would mean that from all the inserts we tested, this one would be one of the highest inserted one, and a bad one would be lower than most of the ones that we tested. So yeah, as long as your insert sequence is roughly what was in our initial pool, it's a really good comparison. So yeah, that's why it's nice to kind of have the distribution of the ones we measured. But you can also try to predict absolute editing rates if you know what editing rate you would usually expect in a pooled experiment that you are running.
Okay, cool. Okay, next one. And I want to remind the audience that if you have a question and you write your name, we will be able to come back to you later if we don't have time today. Okay, so there is another question that it says, notice that there were a couple of data points within the different RTTs that show very high levels of insertion. Was curious if there was anything unique about those particular sequences.
Yes, I think so. So there's one example of a sequence that is through the roof in comparison to all other sequences, which is in the EMX1 target site. And it's quite interesting because that target site consists of, I believe, GAA, 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 GAA. And there was a 3-nucleotide insert GAA that showed up as incredibly high compared to everything else. And yeah, in that case, it's tricky to say if that is a real insert or if that's maybe because there are so many copies of the GAA trinucleotides around it that made this sequence appear like maybe it's an indel or something, but it looks like that sequence. And then I think for some other sequences, there are also specific cases that just where maybe all the feature values are extremely good that could lead to high rates. For example, one of the outlier sequences is even the His6 tag if you encode it completely in the CAC nucleotide. We can't explain all of them, and some of it is probably also down to experimental noise, but I think for some there are explanations.
Perfect. There is a very interesting question, at least I'm curious to know. Are there any technical aspects of your library and analysis that you would have designed differently now in retrospect?
That's a great question. I think we can take probably, I say a few words and Juliana could also say a few words.
Okay.
Yes, so one of them is our library ended up being relatively skewed when we cloned it. And I think that was a purely technical problem. So I think I would change the cloning slightly to kind of not have that skew in there. But then our library, and I think this also answers another question that I saw in the chat. Our library had a few target sites which we cloned separately, so recombination was not a big problem. But we are now doing other experiments where we have multiple target sites within the same library, and we see that recombination, which happens in the lentiviral packaging step, is a problem. So I think if I would try to avoid lentivirus probably in the future if we think recombination is a big problem. We could first generate a landing pad or go piggyback or do some of these things. And then finally, maybe the last point is a lot of improvements to the prime editing system have happened by now. So I would, for basically any experiment, recommend EPEG RNAs. So that's definitely a change we would make. There's some versions of the scaffold that work slightly better. So yes, there is a lot of room for improvement.
Yeah. There's small other things, but I mean, we can maybe focus on more questions because I think you managed them. I mentioned the major points.
Yeah. Okay. Okay. There is one question that it says, well, they're thanking you for the talk, by the way. A lot of people, it seems that everybody likes it. So you use only immortalized cell lines. Do you think that your tools will be also useful for primary or EES cells?
Should I take it?
Yeah.
Yes, I think it's, so I would expect the correlation of the predictions in our tool compared to the results you would get to drop if you move into different cell line context. At least for the base editing and Cas9 tools, we've seen that quite consistently. But I would say that probably some of those features that we discover, like I would suspect length and maybe some of the nucleotide contributions would be relatively universal. But of course, I think the repair processes that are active in stem cells might be a bit different. The way they react to the delivery and to certain nicking and so on might be a bit different. The kinetics might be different. So I would expect correlation to drop, but I think it's better than like nothing as a starting point.
Good. There's one, it's a little bit long, so I will try to read it as clear as possible. So you choose to specifically look at prime editing insertions with this assay. Prime editing can make many different sorts of edits. So first question is, do you think your dedicated insertion library provided particular insights that the general prime editing library, like other labs have used, was not given? And the second question is, do you think similar dedicated libraries for other classes of edits would provide better data on these edits?
So yeah, I think to the first point, our library is very special in a sense that we have a wide variety of inserts that other similar libraries don't have. So for example, the Predict library, which recently was published, they have a really nice data set and model and cover different types of inserts. It's really cool work. But for the different types of insert they're testing, their length is very limited. I think they go up to 10 or 14 nucleotides in length. And our library of 69 nucleotides really allowed us to see different effects for length. And I think mostly because of that, we were able to discover TRX1 and 2, which didn't come up in similar screens before when looking at different repair pathways. So I think for the DPE data is mostly on substitutions, but one type of substitution. And all the sequence-specific effects, I think you can discover them much better by having libraries that have a wide variety on the features and can cover a large space. So I would assume that for small deletions, for more substitutions, the different position effects, there's still a lot of features that are not understood in detail with the current libraries. So having a bigger libraries or sub-libraries, I think, is beneficial for learning more about prime editing and how the editing is happening.
Yeah. I think that we have time for one or more too. So do you use lentiviral transduction only because of a small AAV capacity, or is there any other reason why?
I guess it's very simple. That's maybe one of the, like, we have all the lentiviral plasmids in the lab. It's, I guess, relatively standard for these type of pooled library screens. It's relatively easy to titrate, and yeah, like it worked very well for us in the past. It's just, I think the only thing to keep in mind with lentivirus is they're quite prone to recombination. So as long as we can avoid that, like if either recombination doesn't matter, or yeah, or if recombination is a problem, then maybe can consider other systems.
Yeah, in fact, this can be tied up with another question that there is in the chat. It says that during the lentiviral prep of your guys, did you account for potential lentiviral recombination and potential rearrangement of the sequences?
Yeah, I think it's a very similar question. We sequenced the whole guide actually, but in our case, recombination, we're sort of lucky that it doesn't matter because it happens at the scaffold, and we have the extension and the spacer, but since it's always the same target, the spacer is always the same. So basically, even if recombination happens, you swap the same spacer with a different extension. So in our case, it doesn't matter, but if there were different spacers, it would matter.
Yeah. So I think that maybe we're going to take this one more if you're fine. Okay, do you have a sense from your data which is the longer-lived intermediate, the 3' flap or the mismatch double-strand? Specifically, can you tell whether the increased insertion efficiency from a structure inserts is due to evading TREX or evading MMR?
Okay. That's also a really interesting question. I think I would speculate that it's really interesting to think what's actually going on and why does the length of the insert matter for a flap nuclease. And I assume that for maybe longer inserts, the flap stays probably in the flap state more often compared to in the double-strand state with the mismatches. And maybe if it stays longer in the flap state, that makes it more vulnerable for flap degradation. But it's just a hypothesis. It would be super cool if someone would have an answer on what kinetics are actually happening there. And maybe one more thing about the mismatch repair versus T-Rex. We have this, maybe if you could put up the plot with the length and the heat map.
The heat map.
With the T-Rex, it's in the main one. So it's a bit tricky because short sequences are less prone to form structure, but you can see in that heat map that the structure is more protective for longer sequences compared to shorter sequences, which could in part be because shorter sequences are less prone to form structure. But if it was mainly protecting from mismatch repair, you would expect an almost inverse relationship, right? You would expect that for shorter sequences, structure matters more than for longer sequences. But since it matters more for longer sequences, my hypothesis would be that it's T-Rex related. But again, it's just a speculation.
Yeah, good. Okay, so I think that we are on time. We still have some questions, so we will answer them by email. Thank you so much. It has been an amazing webinar. For the audience, please, you will be redirected to a brief survey. If you have just 1 minute, we will really appreciate your feedback. Thanks, both of you. You have done great, and thanks everybody for attending this webinar. Have a nice afternoon.
Thank you.
Thank you.
Bye-bye.
Bye-bye.
Bye.